Good afternoon, and welcome to our presentation on incellular protein crystallization in microgravity. We are Team 2 from University of Maryland College Park. Presenting today is myself, George Witt, and my colleagues Caio Goolsby and Raina Hatcher. But first, a brief introduction. Overall, we plan to study how does microgravity affect the incellular growth of inosine 5 monophosphate dehydrogenase, or IMPDH, in Spodobactera frugiperta SF9 cells. We also plan to study how this can be extrapolated to the effect of microgravity in general on incellular crystal growth, including, as just some basic examples, does microgravity increase the number of formed crystals in a given sample, increase the size of formed crystals, or increase the order of formed crystals? While we needed to choose IMPDH in SF9 cells as it best fits the requirements for the allowed materials and procedures, we hope to show that this can be extrapolated to the general effect of gravity on crystal growth. But first, some brief background on protein crystallization. Chains of amino acids, or proteins, can crystallize both inside and outside a cell. Studying protein crystallization as a whole dates all the way back to the Brazil knot observations in 1855, though this expanded over following years. It has been since shown that mammalian cells also form crystals naturally and exhibit different characteristics when grown in microgravity, namely larger or fewer impurities, etc., etc., making the process therefore vital to understand for long-term spaceflight missions.
Thus, there is undoubtedly a gap in the scientific understanding of how gravity impacts protein crystallization inside of cells. Our experiment is trying to remedy this by seeing how TB-IMPDH, a variation of the IMPDH protein found in Trypanosoma brucei, a parasite native to Africa, how this protein will crystallize in microgravity in the ISS and on gravity here on Earth. Thus, the results of this experiment will allow us to better understand how this fundamental biological process interacts with long-term exposure to microgravity. If humanity intends to continue expanding its presence into space, this is something we must understand, as protein crystallization is a fundamental part of not only our own microbiology, but the microbiology of the plants, animals, and insects that surround us on a daily basis. In order to do all of this, we propose to send up a Type 3 FME mini-lab to the International Space Station. In the first volume of this, there will be a concentrated baculovirus stock in a nutrient solution. The nutrient solution's composition will have to be experimentally determined later to optimally extend the lifetime of cells. The virus stock will either have to be developed in-house or contracted out to Thermo Fisher to genetically modify it so that infected SF9 cells will express TB-IMPDH. At U-14, this volume will be then introduced to Volume 2 by unlocking Clamp A, which will include a cell culture medium of the SF9 cells. At this point, the SF9 cells will become infected and develop the protein crystals. At U-5, Clamp B will be unlocked to expose the entire solution to Volume 3, which will be a concentrated phosphate buffer, which will stabilize the protein crystals so that they will survive the descent down.
Once we have reobtained our microgravity experiment from the International Space Station, we will split each FME sample, including the control sample, in half to be analyzed. One half of each sample, control and ISS, will be imaged through a transmission electron microscope, which will determine the quantity of crystals in each cell. Then, the second half of each sample will be analyzed through X-ray crystallography, which will determine the resulting crystal structure and durability using the global distance test. These different metrics will give us a more well-rounded view of the effect of microgravity on incellular protein crystallization, and also give us a more versatile manner to study the cells in case one method is unsuccessful. And then we come to our works cited. Thank you for considering our experiment, and we hope you enjoyed our presentation.